Okay, so today we'll begin with bioinorganic chemistry. In the demo class, I told you about the units that we are going to discuss in inorganic chemistry. Bioinorganic chemistry is one of the most important topics. And I told you at CSR level, what is the maximum mark that we got at CSR level, if you remember? The maximum mark that we got is 16 marks. So this is the maximum mark that you can get from this unit. 16 marks only from one unit. Apart from this, even if you go for GATE exams or SET exam, whatever may be the exam, as I told you, they have to definitely give a question from bioinorganic chemistry, or you call it as a bio-coordination chemistry. Okay, so this is a very, very important unit. So concentrate, and whatever notes that has to be given, I will be dictating you. You have to just note it down, all the important points. But this is like, how many of you are from life science background? 1, 2, 3. Okay, you are from MPC background? MPC background. Okay, no issue. So absolutely no problem for MPC students, because we are going to study only chemistry related to this biology. Okay, not even biochemistry. We are not going to study anything about life science directly, only a chemistry related, and especially the coordination transition elements, how they behave, what are the ligands that are present around the metal. That is what we are going to discuss. So for this reason, bioinorganic chemistry is also called as bio-coordination chemistry. So it is the coordination chemistry at biological level. To say simply, it is the coordination chemistry at biological level. Okay, so put the main heading as bioinorganic chemistry or bio-coordination chemistry. Very important topic for every exam. You just name an exam, they will definitely give one question related to this. Even at JAM level, they give a question from bioinorganic chemistry. Write the definition. It is the study of coordination chemistry. It is the study of coordination chemistry at biological level. It is the study of coordination chemistry at biological level. Can you name any scientist? Okay, can you name any scientist who got a Nobel Prize in biological sciences related to oxygen binding proteins? Oxygen binding proteins, or specifically I'll give you related to myoglobin or hemoglobin. Scientist who got a Nobel Prize. Okay, we'll see. But before getting into that, you just look at this famous picture. You might have identified what is more important about this picture. Yes? Alexander Fleming. This is related to chemistry. Conference related to quantum chemistry or quantum mechanics, quantum physics, everything related to quantum. Can you identify at least one or two scientists? Maybe, yeah, Einstein, right? Pictures are not that clear, it will be little blurred. Planck is also there, right? Can you identify this person? Niels Bohr, right? Very good. Because you are able to identify the scientist. This one? A very big equation in your quantum chemistry. Dou psi x, dou psi y, dou psi Schrodinger. So this person is Schrodinger. And what is unique about this picture? There are 27 scientists. Out of them, 17 of them are Nobel laureates. Anything which is different, looking different in this picture? Anything which is looking different? Pick the odd man out. General aptitude. In part A also sometimes question appear. 6, 8, 10, 9. Pick the odd man. 6, 8, 10, 9. Which is odd? 9 is odd because all the three are even numbers. So just like that, pick the odd in this picture. Can you see a women scientist? Only one women scientist in this. Yes? Marie Curie, Nobel laureate. She finally died of cancer because she worked on nuclear chemistry. Mostly she worked on nuclear chemistry, and whatever radiation that every time used to come, she finally died of radiation, the effect related to cancer. So this is a picture. They have modified it and they have given every name. Almost you can find every name related to quantum mechanics. Dirac, have you heard about Dirac? There is a famous equation of Dirac's where it says that everything will come in doubles. Everything come in doubles in the sense, suppose if there is anywhere positive in this universe, there has to be negative in the universe, definitely. And it goes on to say that suppose if there is matter, suppose that if there is matter in this universe, there has to be antimatter in the universe also. There has to be antimatter. So it went on to say, suppose if I am Shashikant, there should be some anti-Shashikant anywhere in this universe. Definitely he has to exist. Or whatever that I am there, the anti of that should exist. So that is a proposal came from this person called as Dirac. He has given Dirac equation. You can see Einstein, Curie, Max Planck, Langmuir, everyone. But anyway, coming to bioinorganic chemistry, there are two scientists, John Kendrew and Max Perutz. These are the two scientists. These people have discovered the structure of myoglobin. Whatever hemoglobin or hemerythrin, hemocyanin, everything that we are studying about oxygen binding proteins, the most important work was done by these scientists. Max Perutz, we call it as Max Perutz, and John Kendrew. And you can see all these are molecular models. They used to prepare molecular models. All these are like carbon tetravalency. So they used to connect them and then they used to check. They used to prepare all the adenines, guanines, and then they used to connect them. So whatever DNA that is present, they used to prepare that DNA or adenine, guanine. Because suppose if I say hemoglobin, what does hemoglobin contain? Iron surrounded by porphyrin, which you call it as a heme portion, but it is also attached to a protein chain. That protein chain will contain certain amino acids. Protein chain will be having some amino acids, and all this is related to some nucleic acids also at a higher level. Okay, so that is what they are doing. Right. What is the basis of life? This is a very interesting unit because it is related to life sciences. What is the basis of life? Suppose if I want to survive, what should I do daily? I have to eat or I have to drink something, otherwise I will not survive. Where is that coming from? Maybe if I am a non-vegetarian, I will be eating meat daily, or if I am a vegetarian, I will be eating vegetables. Where are these coming from? What is the source for these non-vegetarians? The source is, say, some meat, or let me give an example, a sheep or a goat. But how do they survive? They survive on plants, grass, plants, everything, or vegetation. They survive on vegetation. But how is vegetation surviving? What is the basis for the vegetation? Every day I don't go and pour water. Only in my home, if there are any plants or trees, then I will be watering them. But the regular water anyway comes. What is the other source? Other source is? Yeah, but who is giving all this energy? Sun. That is what is the basis of life. So basis of life, we feel that it is something else, but sun is the basis of life. Because it is the photons or the rays that are coming from the sun that will be taken up by the chlorophyll. They get excited by taking up these photons, and once they get excited, they are highly unstable. They try to release electron, and whatever electron that is released, that electron is at a very high energy. That high energy will be given off, and that will give you the processes of photosynthesis and so on. So whatever energy that is coming out, it is trapped in the form of ATP. That ATP will again further go into dark reaction, or which you call it as Calvin cycle, and then it is converted into ADP, where carbon dioxide and water are converted into carbohydrate, or simply carbon dioxide gets reduced into carbohydrate. Water gets oxidized into oxygen, and it will give away oxygen. That is what we are inhaling and we are surviving. So it is just like a simple reduction and an oxidation type of reaction, photosynthesis, where carbon dioxide is getting reduced into carbohydrates and water is getting oxidized into oxygen. Okay. But we say that everything needs light, but here is a fish. This is in a very, very deep sea, where light cannot even pass. So deep sea animals that doesn't require light, but still it survives. Without light also it can survive. So whenever we say something in chemistry especially, there will be an exception. I say about anything related to coordination chemistry, immediately some exception arises. So here also it is like an exception, where without even light it is able to survive. This is one such organism. Can you identify this? One of the models of periodic tables. But anyway, this is just introduction class. All these are different elements. But can you name the element which is abundant in us by weight? The element which is abundant by weight in every human being. Iron, it is anywhere about 5 grams altogether for an average human being. Whenever in bioinorganic chemistry, remember, whatever figure that we give, if I say 5 grams of hemoglobin, then there is a ideal body. Ideal body should weigh 70 kg. For a 70 kg human being, for a man, there should be 5 grams of hemoglobin. If it is 60 kilos or 50 kilos, that will vary. But always the average weight has to be 70 kg. So whatever they describe, that is related to that 70 kg man. Okay, not iron. Magnesium, no. Magnesium is very less quantity we require. How much magnesium do you need every day? Magnesium is very good for muscles. It is good for our bone strength also. Everything, it is related to everything of our body. Magnesium. Myosin. Yeah, I am talking about an element. Magnesium is oxygen, because nearly 60% of my body weight is water. What is water constituted of? H2O. And in H2O, there are 2 hydrogens, 1 oxygen. So we are not speaking about how many elements are there, the abundance by weight. By weight means oxygen will always outweigh, because it is 16. Molecular weight is 16, hydrogen is only 1. 2 hydrogens, but molecular weight, I am just saying it is only, if I take it as 1, then oxygen is 16. It is 16 times heavier. So which is the most abundant element by weight means it is oxygen. It is making up about 53% of the weight. So half of my weight is just because of oxygen. Half of your weight is because of oxygen. Then comes carbon. Okay, so just add a point. In some set exam questions, general knowledge questions, these things will come. Write a point. The basis of life is sunlight, which helps in the process of photosynthesis, which is the basis of further life, which is the basis of further life. Next point. Human beings are the most important species with life. Next. The most abundant element by weight percentage is oxygen. In bracket you just write around 50%. They give it as 53.6, but on an average you have to just write 53%. Continued by carbon, comma, hydrogen and nitrogen. Carbon, hydrogen and nitrogen. Okay. So you just remember 4 important elements. What are they? Anyway, we are organic chemistry students. Most of you are organic chemistry. So we start always from carbon. You remember CHON, Chon. Right? You might have heard about Jackie Chan. You remember Chon. CHON, carbon, hydrogen, oxygen, nitrogen. So even in the organic chemistry, always it is like more amount of carbon, then will be hydrogen, then will be oxygen, less will be nitrogen. You'll also have some heteroatoms like sulfur, phosphorus, chlorine and so on. But these are the 4 important elements. Okay. Apart from this, do you have any other elements in our body? So when I'm speaking about life in bioinorganic chemistry, 99% of the time I will be speaking about human beings. Because different elements will have, different organisms will have different element constitution. So generally I'm speaking about human beings. Apart from CHON, do we have any other element? Calcium is present, otherwise I will not survive. No bones, so no skeleton. Calcium is there. I already got 5 elements. Still, just now we spoke about magnesium. Magnesium is also one of the elements which is very, very important element. How much magnesium do you require per day? Calcium anywhere about 500 mg. Some people may require 1000 mg also. 1000 is the maximum limit, but usually 300 to 500 mg is on average. Every day if I don't get that, my bone strength will not be very good. My heart health will not be very good. Heart health is also related to calcium content. There are something called as calcium channels. These calcium channels will be regulating calcium content, which will maintain the health of the heart. So if you are not intaking proper calcium, that can lead to heart diseases also. Nobody concentrates on calcium. Everyone concentrates on cholesterol and so on. But calcium is also very important. Magnesium we require approximately 300 to 400 milligrams per day. But how do you get that? Vegetables, yeah, that is one source, because whatever vegetable I eat, mostly it will be like green leafy vegetables. That green leafy vegetable will have chlorophyll. That chlorophyll will contain magnesium. So through that I will get magnesium. But that is not to my required quantity. Still I want more magnesium. Where can I get it from? Vegetable is one source. The other source, there is one of the best sources of magnesium, which is very cheaper also. It also gives you protein. It also gives you protein, but it gives you good amount of magnesium. That is very important. You eat dosa idli. Along with dosa idli, what will you take? Chutney. Generally in chutney, what do you have? Ingredient? Mostly it is like peanut. Peanuts contain good amount of magnesium. And every day if you take good amount of peanuts, that will provide the protein requirement, and also it will provide the magnesium requirement. So magnesium is also very, very important. Right. Can you name two more elements at least? Potassium, right? Chloride, sodium. Now you are increasing. Almost we have come up to 10. There are nearly 29 elements in our body. Our body is made up of approximately 29 elements. Nearly you have told 9 elements. It means there are 20 more elements. So sodium, chloride, potassium, these three maintain osmotic potential. Still, you have mentioned iron. Okay. Now the question is, you just name a few transition elements. Transition elements which are present in us. Name a few transition elements which are present in us. Zinc, right? Zinc is present. Most of the metalloenzymes contain zinc. We have very less amount of zinc content. It is lesser than iron. But suppose if I give you 100 metalloenzymes, 70 metalloenzymes will contain zinc at the active site, not iron. Zinc is more important. But the sad thing is, availability of zinc is very, very low from the foods. So most of us will be like zinc deficient. Suppose if you have any fever, the first thing that you do is, I don't want to eat. You don't like to eat. Or I'm eating but I'm not getting any taste. Then you go to a doctor, he'll give you a syrup. What syrup does it contain usually? Zinc related syrup. So once zinc is supplemented, again your hunger will also increase. Taste will come. So everything is related to zinc, very, very important. But zinc will be very less. So you can get it from dry fruits, dry nuts, dry seeds. That is one source. The other source is meat. Mostly zinc can be get from meat. And the richest source is oyster. It is a seafood, oyster. That is the richest source of zinc. Okay. But anyway, other elements, any transition elements that you want to mention? Copper, right? Cobalt. So you are coming from below. Zinc. Then you said copper. Nickel. Nickel is also found. Then you said cobalt, iron, manganese is also present. Still, scandium you don't find, titanium you don't find. Vanadium is found in some organisms. Chromium is also found to some extent. So if you take 3D transition series, apart from those, the first beginning 3 elements, almost all elements are present in us. Okay, and if you just mention any element, can you mention an element which is beyond 30 atomic number? Beyond 30 atomic number. Any element which is beyond 30. Because whatever elements you are seeing here, all these, they are under 30. Nitrogen, sodium, potassium, sulfur, chlorine, silicon, iron, fluorine. Fluorine is also under 30. Chlorine is under 30. Zinc is under 30. Copper is under 30. Bromine beyond 30. It is beyond 30. Selenium, arsenic, lead, all these are like beyond 30. So very few elements are there beyond 30. Okay. Bromide, where do you get it from? Some amount of bromide. In fact, it is good for our, suppose if there is any person who gets cough, then there are some syrups which will contain some bromides. The cough gets subsided. Instead, you can also eat a fruit. From that fruit also you will get the bromide. What is that fruit? That can subside your cough. What is that fruit? You have heard about pineapple? Pineapple contains bromide content. So that can be used for relieving some coughs. Okay. Anyway, put a small statement, write a small statement. Human beings approximately contain 29 elements. 29 elements. Next. Most of these elements have atomic numbers. Most of these elements have atomic numbers below 30. Below 30. Next point. These elements are classified into metals and non-metals. Metals and non-metals. Metals and non-metals. These are write down this classification. In set exams, they'll ask a question directly from this table. They'll ask which of the below is a trace element. Trace means, suppose if I am taking in a greater quantity, I call it as macro quantity. If it is lesser quantity, we say micro. If it is still lesser, we say trace. If it is still lesser than trace element, we call it as ultra trace element. So here you can see these are macro. Macro means sodium, potassium, calcium and magnesium. These are found in a good amount of quantity. Next one is trace element, iron, zinc and copper. Ultra trace. Ultra trace means their quantity is very, very less. In this, if you are carefully watching it, vanadium, chromium, manganese. Vanadium is also found. I told you vanadium can be found in some organisms. Chromium is found. Manganese, cobalt, nickel. Molybdenum is above 30. Lead, arsenic, tin and cadmium. Lead, arsenic, tin and cadmium, generally they should not be found in our bodies because they are like toxic. They are not good for our health. But these are also included into the classification. The reason is the food content that we are taking, that is giving some amount of these things. So that is also included as an element. It is going and sitting in our body. How is lead coming? Through lead pipes or whatever the cans, the tins that are made up of. Through that lead is also slowly seeping. But once lead goes into my body, what will happen? It will replace calcium. Wherever calcium is present, immediately it will go and sit in the place of calcium. That calcium will come out. And where do that calcium go? Into the body. So calcium content in the body will increase. So that will disturb the calcium channels. That will disturb the heart health. That is one. Second thing, if there is more calcium in my body, it has to pass through the blood. Then again it has to get filtered into my kidneys. So now kidneys have got little bit of calcium. Every day, due to my health consciousness, I'll eat leafy vegetables. Leafy vegetables will have oxalates. They have good amount of oxalates. So this calcium will combine with oxalate, that will form calcium oxalate crystals. What are these crystals? You might have heard about kidney stones, calcium oxalate crystals. So if that calcium is coming out from the bones, it will be into the blood. From there it will be into the kidneys. From there it can combine with oxalates and it will give you calcium oxalate, which you call them as kidney stones. So you have to drink more amount of water. That is the reason they say drink more amount of water, so that oxalates, it will not combine with calcium. Anyway, you first note down this. I have classified them into metals and non-metals. Metals, macro and micro. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur and chlorine, these are called as macro, non-metals. Boron is also there. Boron. Silicon, selenium, fluorine, iodine. Selenium containing foods are very rare. Some multivitamins, it contains selenium also. But remember, the last 4 elements, they are generally not essential elements. Essential elements means the elements which are required by our body. These are not essential elements. Lead, arsenic, tin, cadmium, they are toxic. Some of you might be taking soft drinks not from the glass bottle, from the tins. So there will be metal. That is, the tin is made up of metal. And wherever metal is having some acidic nature, that metal will seep. And whatever soft drinks that are there, how do you maintain the soft drinks for 6 months preservation? Soft drink contain sugar. Definitely it will contain sugar. Without sugar there is no soft drink. Once sugar is there, there will be microbes. But microbes should not come. Then how should I preserve it? Carbon dioxide is not a definite ingredient. If carbon dioxide is there, we call it as carbonated beverages. But maybe there is a fruit drink. That fruit drink will not contain any carbon dioxide. They will not put any carbon dioxide, just fruit juices. So if you open them, you will not get any gas. But carbonated beverages, if you open them, immediately gas will come out. That carbon dioxide is dissolved for taste. Taste in the sense, whenever I dissolve carbon dioxide into water, what does it form? Carbonic acid. So once you are drinking it, there will be like a tingy taste. Tinginess will be coming on your tongue. That is the reason they put carbon dioxide with force. So once you are opening it, and if you shake it and open, immediately that carbon dioxide will fuzz out. So it will completely come out. Finished this one? The ingredient generally they add is acid. One such acid is phosphoric acid. In cool drinks they also add phosphoric acid. So once they add acids, the pH of these soft drinks will be anywhere between 3 to 5. So they are very acidic. It is not neutral. They will be very much acidic. 3 to 5 you can imagine. So once it is 3 to 5, the seepage of metal will be more. You might have heard, suppose if you are cooking, what is the common metal that you are cooking at home? Aluminium. Aluminium is the most common one, but they say aluminium is also not good for health. So you may replace it by steel or maybe some earthen pottery, some pot containing one. Or I generally don't use brass. I generally don't use copper. What is the reason? Brass is very good, but it is generally for storage. For suppose if you are preparing a curry, maybe you might have seen your grandparents, they don't prepare curry in brass utensils in general. They can prepare food. Rice they can prepare, but generally not curries. What is the reason? Because if I am preparing some curry, there is some tinginess agent which we are adding it. Maybe I will be adding tamarind. Tamarind you will be adding, right? Or maybe tomato you will be adding. Some vitamin C contents will be there. Or in tamarind what do you have? Tartaric acid will be there. So in the presence of these acids, metal will seep. Metal will come. So that will give you more copper. What does brass contain? zinc, copper. So more amount of zinc will come, more amount of copper will come. In little quantity it is good for our health. Maybe you might have heard about Tata Copper. Have you tasted Tata Copper water? It will not taste like normal water. It will be like little bit different taste. Because they add these metals into it. Tata Copper Plus, they say. They'll be adding little metals into it that will change the taste also. Because metals are present in it. This is not required to note down, but just have a look at it. Required daily dosage. Potassium, approximately I require about 5 grams. They have given it in milligrams. 4700 milligrams. Sodium is also very high. Calcium is also very high. Anywhere between 100 to 1300. That depends. Maybe if there is a pregnant woman who has delivered, their bones need to be more stronger. They want more calcium. So for that reason, it can range up to 1300 also. It is average 100 to 1300. Magnesium 310 to 420. But just look at zinc. Just 8 to 11 mg. The required quantity is very, very low. And whatever quantity that comes into us, most of it directly goes into metalloenzymes. It will not be stored anywhere else. Directly it has to go into metalloenzymes. But iron is there. Iron anywhere about 5 grams is required. It can be stored. It is stored. All those things also we have to discuss. This is the concentration of sodium, potassium millimoles. How much sodium is present, how much chlorine and how much potassium you can see. Sodium and chlorine is very high, but potassium will be low. So potassium content will be low in this. This is an organism which can store which metal? Vanadium. So there are some organisms which can even store vanadium. Vanadium is required in very, very lower quantities. But this is one organism which can store it. Okay, now put a heading. Physiological effects of metal concentration. Physiological effects of metal concentration. Write a point. Any metal present in the human body, any metal present in the human body should be at optimal level. Should be at optimal level. At optimal level. Decrease or increase leads to metabolic issues or metabolic problems. Decrease or increase leads to metabolic issues. Then note down this diagram. One is the concentration which is required for the survival. If any person has to survive, this is the minimum quantity that is required for us. Otherwise the person will die or the organism will die. What is 2? Deficiency. It means apart from my survival, I have to survive, but along with that survival, whatever metal or whatever element that is required, it is not at its optimal level. It is little bit less. So it is like deficiency. Suppose if I've got hemoglobin content anywhere about 15, then I'm not deficient. But as a man, if I got anywhere around 10, then we call it as deficient. Then you can call them as anemic. For women it should be anywhere between 12 to 14, but for men it should be about 12 to 17 and 18 also. 17 is okay, maximum. So if it is less, you call it as deficient. And if the metal concentration is still increasing, then that reaches at optimal level where all the physiological activities will be good. This is a physiological effect, it will be good at optimal level. Suppose if the metal concentration is still increasing, someone says iron is very good, it maintains your hemoglobin levels. Then if I start eating iron more, then it will definitely harm. So if the concentration of that metal is increasing, slowly it becomes toxic. If the concentration is still increasing, it becomes so dangerous, lethal, that the person may die. If the metal concentration becomes very high, the person may also die. Can you name a metal which was ingested by some people who died by eating some fish? It happened in Japan. That is a very famous GK question, and also it has appeared in many exams. Chemistry related exams it has appeared. What is the disease called as, and what is the metal that was ingested? Which metal they have taken? It comes in many competitive exam questions also. Finished? Have you written all those things? 1 is survival, 2 is deficiency, 3 is optimum, 4 is toxic, 5 is lethal. This is like a GK for you. Minamata disease. Mercury, right. Industries, whatever effluents that come out of industries, that should be treated in a plant. But instead, what some people will be doing is, they will be just leaving all that effluents into the waters, rivers, or the other waters that are taken up by us. And those waters will have very high content of toxic metals. And that water had mercury, so that fish was, anyway, it will be eating some organisms present in water. Slowly that mercury also went into its body. Mercury was in the fish. And that fish, Japan people, mostly they rely on seafoods. So that was eaten by some people. They got a disease called as Minamata. They died. Later they came to know why they died. The reason is mercury. How did mercury come? Then they went on to investigate. This mercury has come from the fishes. How did fish got that mercury? From the industrial waste that was letting out into waters. So who should be more responsible in this world? Most of us, but who should be more responsible? We should be more responsible. We, or mostly you, should be much more responsible if you are entering into pharma or industrial field. We are anyway in the teaching field. Maybe in the future if you are going into research field, or if you are in industrial field. So if you are at a very higher level, maybe you are in production, then you will say effluents treatment will require some money. Every day whatever industrial effluents that come out, you have to send it to the treatment plant. Either you have to maintain it or you have to send it. Every day tankers will come, it will take the effluent and then they will treat it. Then the solvents will also come out, they can distill it. But suppose there is a rainy season, then you want money. You will say, why to invest money by treating the effluents? Simply leave it. Rain is anyway coming. So leave it through the rainwater, it will go. So it will go into homes, it will seep, and that will lead to all issues. That has happened many times. Anyway, this is, you might have heard about dimer-caprol. Dimer-caprol, why is it used? This is generally a question at MSc entrance. Okay, write a question. The ligand used to bind some toxic chemicals. The ligand used to bind some toxic chemicals like mercury and arsenic is write down the ligand dimercaprol. IUPAC name is also written 2,3-dimercaptopropanol. Dimercaprol. You call it as BAL. BAL. Next one is BAL. That is also used as a ligand. What is BAL? British anti-lewisite. Dimercaprol. BAL. Penicillamine, EDTA. All these are used to bind with some heavier metals. EDTA is also one of the most important ones. Suppose if calcium is very high in my body or lead is very high in my body, then they will simply administer EDTA to us. That EDTA will go and bind to that excess calcium or lead and it will remove that. If this point is completed, note down this point also, because these things come in the exams. EDTA is used to bind metals like calcium and lead, which are in excess. What is EDTA? Ethylene diamine tetraacetic acid, but generally you will not get it as ethylene diamine tetraacetic acid. How do you get it as? In the laboratories, directly you don't get EDTA as ethylene diamine tetraacetic acid. You will get it as a salt. What is that salt? It is sodium salt of EDTA. So you call it as sodium salt of ethylene diamine tetraacetate. You call it as acetate because you will get it as a salt. And which salt is it? Sodium salt. Note down. EDTA mostly comes as sodium salt. So it is sodium salt of EDTA that we generally use. Okay, now a general question. Because this is like introductory class, mostly it will be like questions. Explanation in the beginning will be less. Mostly it is like an interaction. Now the question is, can you name a biological ligand? You are aware of ligands? Ligand is anything that can go and bind with a metal. Okay, I can give a simple fluorine, I can give a simple chlorine, that can also act as a ligand. Aqua can act as a ligand, amine can act as a ligand, plenty of ligands. When we discuss about coordination chemistry, I'll give you a list of about 100 ligands. Out of those 100 ligands, you have to remember at least 50 ligands for your examination point of view. I'll tell you which are the important ones. You have to remember those 50 definitely. But I'll give you a list of about 100. I have 100 ligands. But here my question is, at biological level, can you name a few ligands which can go and bind to the metal? Lithium, no. Lithium is a metal. Lithium is not there in us. Suppose if there is any lithium in us, it is dangerous. Where do you find lithium? Mostly where do you find it? Everyone is having it. Mobile. So lithium batteries. Mostly lithium is found in batteries. But that lithium is also generated in our bodies through a therapy, not regularly, through a therapy. Suppose a person gets cancer. That cancer can be treated by radiotherapy, chemotherapy and so on. But there is a therapy called as BNCT, boron neutron capture therapy. So a cancer patient will be given boron. Once they give boron, then they will bombard the boron with some neutrons. They will bombard boron with some neutrons. Which boron? There are two borons, B10 and B11. Two isotopes are there. So one of the isotopes is active. And that boron, after bombarding with the neutron, that will give you lithium and helium. Helium means alpha particle. And that will destroy the cell. So lithium formed will destroy the cell. Helium particle will also destroy the cancerous cell. That is one treatment. We call it as BNCT, boron neutron capture therapy, used for cancer treatment. But anyway, I want any other ligand. Water at biological level. Okay, water is one ligand. Apart from that, I want little larger ligand. Every one of you have completed your masters? Anyone pursuing? You have completed. Okay. So bioinorganic chemistry was there? It was there. For some, maybe they don't include bioinorganic, very few, but almost every university will have bioinorganic chemistry. Very important topic. Porphyrin is a ligand. If I talk about hemoglobin, what is the metal? Iron. But what is the ligand? Porphyrin. Porphyrin is the ligand. It is a ligand. You take it as a ligand, okay? So that is called as a macrocycle. It is not a simple ligand. It is a very large ligand. That is a cyclic ligand. And it is a large cycle. Therefore, I call it as a macrocycle. Those can act as ligands. Those can act as ligands. Okay, that is one. Any second type of ligand at biological level? Most common ones. Adenine can act as a ligand. Your nucleic acids can act. That contain adenine, thymine, guanine, cytosine. Okay, these can also be taken as ligands, right? One most important part you are missing. Macrocycles we got, adenine, guanine, this also we got, where the nitrogen goes and binds with the metal. One more. Very, very important at biological level. Oxygen, no, it will bind as a ligand, but for say hemoglobin, it will go and bind at hemoglobin. That is one example. But other than that, pyrrole. Okay, you are coming little closer. Let me put it this way. These are amino acids. Okay, so there are plenty of amino acids which act as potential ligands. Proteins. These proteins are made up of amino acids, and these amino acids will come and bind with the metal. Suppose if there is a metal, whatever protein that is present, that protein will not be like linear. That protein will be like a chain form. So all that protein will bend. Whenever it is like bending, it is making different forms. The amino acids that are coming, that will form a particular geometry. Without metal, remember, in coordination chemistry, whenever I give a metal and if I add a ligand, then geometry is formed. But at biological level, the geometry is predetermined. Suppose if there are amino acids of a protein, then this is one histidine. One more histidine will come from here. Third histidine, fourth histidine. There are 4 histidines. Inside there is a space. Into this space, if some metal goes and sits, that becomes a metalloenzyme. So another potent ligand at biological levels are amino acids. For these amino acids, mostly nitrogen and oxygen. Sulfur is also there, but most of the ligands will contain nitrogen and oxygen as the donors. Can you name any amino acid which is acting as a ligand with nitrogen as a donor? Or oxygen, anything from your guanine? It is not an amino acid. I want an amino acid. Guanine is not an amino acid. You should have an amine group and an acid group. No. Just now I said one is histidine. Histidine is one. Glutamic acid, aspartic acid. All these can act as ligands. Glycine. Glycine can also act. Tyrosine. So these are some amino acids which can act as potential ligands. Okay. So write the next statement. At biological level, ligands are distributed in different forms. At biological level, ligands are distributed in different forms. These are, note down these 3 points. One is amino acids, one is macrocyclic ligands, and the another one is the bases, adenine, guanine, and so on. Any macrocycle example? Just now we discussed iron is surrounded by porphyrin. I'll give you the examples also, most important examples, and many times asked in the exams also. Directly you have to remember the ligand and the metal generally surrounded by that ligand. And remember one more point, at bioinorganic chemistry, at examination level, every point is like a question. Every point. At CSR level, whatever statements that we give, theoretical statements, directly those theoretical statements will come in the options. So mostly it is like a theoretical bioinorganic chemistry. Your organic chemistry, every time you have tried reactions, whatever topic you choose, definitely there will be some reaction. But at bioinorganic, number of reactions are very less. Photosynthesis, you find a few. Finished? Nucleic acids with nucleobases as potentially coordinating components. Example, adenine and guanine. These are just a few amino acids that I am showing. You need not note it. It will come in your organic chemistry. Histidine, the most common ones. Okay. Why the biological level will prefer mostly nitrogen at its active site? Why nitrogen containing donors? Oxygen containing donors are also there, but less in number. Nitrogen containing donors are very high in number. What could be the reason? What could be the reason? At biological level, even you go to metalloenzymes, these metalloenzymes will have one nitrogen containing ligand, and then it could be oxygen or sulfur or something else. But always you find some nitrogen containing amino acid. What could be the reason? Anyone? Histidine is the most important amino acid. Why nitrogen? I'll give you a clue. HSAB principle. That is the clue for you. Hard base. Nitrogen containing ligands generally are not hard bases. What are they? Yeah, it contains lone pair. Only one. You are coming little close, not hard base. Little closer. Nitrogen containing ligands are borderline bases. They are borderline bases. They are not very hard, they are not very soft. So once it is going and binding to the metals, most of these metals are transition metals belonging to 3D series. So these are like what do you call manganese, or say iron, cobalt, nickel, copper, zinc. All these are generally present in +2 oxidation state. So when I take them in +2 oxidation state, let it be iron, cobalt, copper, nickel, or even zinc, all these present in +2 oxidation state are considered to be borderline acids. Since these are borderline acids, which ligand do they prefer? A borderline base. Which amino acid is like borderline? Nitrogen containing amino acids, especially histidine. So that is one reason you will always find some nitrogen containing ligands. In hemoglobin, you said iron is surrounded by what? Porphyrin. Porphyrin contains what? Porphyrin contains 4 pyrrole rings. The pyrrole ring will have nitrogen. So mostly it will be like nitrogen environment. Understood? Note down. At biological level, nitrogen containing ligands are very common. Nitrogen containing ligands are very common because these are considered as borderline bases. Because these are considered as borderline bases. Example, histidine. So whenever I say histidine nitrogen, every time you don't write all these things, there is a shortcut. We write it like this: capital N, the nitrogen donor atom, and subscript his. It means histidine. So HIS represents histidine. Okay. Suppose if I write cis, then suppose if I write S cis, not CIS cis, not cysteine. This refers to cystenyl sulfur. You can just note down N histidine, nitrogen we abbreviate it as N his, cystenyl sulfur is abbreviated as S cis. These are abbreviations, you have to remember. Tyrosine, TYR. What is the donor atom? Tyrosine. Okay, you write one thing, you write histidine, you write N his. You write methionine, S met. Cysteine, S cis. Selenocysteine is very rare. Tyrosine, O TYR. Aspartic acid and glutamic acid, OASP, O-glue. The name of the amino acid should be like a subscript, abbreviated subscript. Where should you write the subscript, superscript, whether it should be right or left, that is also important. Because if I ask you 6 carbon 12 isotope, how will you write? Carbon Where will you put 6? Can I write it like this? Your isotope, carbon isotope, 12. This is wrong way. I should not write 6 here. Where should I write it? Left subscript. Is it right now? This is also not correct. This is the right one. 6C12. Both should be towards left hand side. Atomic number and atomic mass should be towards left hand side. One should be subscript, one should be superscript. In your old books, you will find 12 here. They are old books. But according to IUPAC, you have to write it towards left hand side. If at all there is any charge, maybe if there is any charge or you have removed an electron, that should be indicated here. That should be like right superscript. Okay, but not here. And how many number? 4 carbons. Then I write it as C4, H10, so on. This is right subscript. So where you are using also, that is important. Okay. Histidine can generally bind with which metals? Methionine can generally bind with which metals? This is, you just write down these 3. You write first 3. These are generally asked in the exams. Which of the below metal can easily bind so and so amino acid? This is purely theoretical. So once you understand, directly you apply, knowledge based. You don't have to do any problems also. That is the reason many students will concentrate on bioinorganic chemistry, because it is marks fetching. You have to just remember them. Understand the concept. Remember the points. Iron 2, 3, copper 1, 2. Zinc, copper 1, 2 or iron. And cysteine with zinc, copper, iron, molybdenum and nickel also. Here very rare nickel can be found in +1 and +3. Because generally you will find nickel in +2. But rarely you can find nickel in +1 and +3 also. Those oxidation states are given here, +1 and +3. Tyrosine with iron 3, it will come in further classes, it will come. Yeah. Now put a heading. Macrocycles found at biological level. Macrocycles found at biological level. Next point. Write a point in that. The four most important macrocycles at biological level are first one, porphine or porphyrin. Porphine, chlorin, not chlorin, there is no E, just chlorin, CHLORIN, chlorin. Third one, corrin, CORRIN, corrin. And fourth one is corphin, CORPHIN. Porphine, chlorin, corrin and corphin. You have to draw them. In the exams, how the questions have appeared previously, I am telling you. The metal generally found with porphine or porphine microcycle has this metal? Any answer? Porphine microcycle is generally bound with this metal. What is that metal? Which metal generally does it binds with? Porphine. Iron. One of the best examples is hemoglobin. Iron. Even in cytochromes you will find iron. Okay. Chlorin. Magnesium. And the pigment. Two questions appear. One is they ask what is the protein or the pigment. The protein here is hemoglobin. One example that I can give you is hemoglobin or myoglobin or cytochrome, so on. This one, chlorophyll. So which of the below rings is found in chlorophyll? Porphine, chlorin, corrin, corphin. Answer is chlorin. Next, where do you find this? Cobalt at the active site. So where do you find it? Where do you find it? Not at cytochrome. Cobalt is correct. Okay, you will get the answer immediately when I attach this. I have attached cyanide. So what could be it? It is attached to some amino acid. It is actually attached to some benzimidazole derivative at one side. Cyano cobal amine. Cyano cobal amine. Coenzyme B12. In coenzyme B12, which of the following rings is present? Answer is corrin ring is present. You don't write the metal, first draw the macrocycles. Later I will give you those points. And corphin, it is found with some nickel containing proteins. Corphin. We call it as hydrocorphin also, because you can see there are more number of hydrogens. One more question that they have asked is how many double bonds are in conjugation in porphyrin? That is also important. 11 double bonds will be in conjugation. Altogether how many electrons are involved in conjugation? 20, 22, 24, 26. How many pi electrons or how many electrons are involved in conjugation? 20? Yeah, 22 is one answer. Suppose if there is no 22, they have given 18, 20, 24, 26. 18, 20, 24, 26. 22 is not there. Then is it a right question or wrong question? Options. So you find 11 double bonds in conjugation. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 double bonds. 22. These lone pairs also participate in conjugation. So 22 plus 2, 24 plus 2, 26. If there is no 22 option, the next option has to be 26. Okay. Anyway, first you note this. These examples I'll give you later. First you just draw these structures. The easier way to draw them, first of all you draw the first two. The easiest way I am showing you. First you put 4 nitrogens like a square. Okay. Once you have put 4 nitrogens, make them into pyrols. Four pyrols are done. Then join them with methane bridges. Done. So easily you can draw it. So put 4 nitrogens, make it part of pyrrole rings, put 4 methane bridges, and then double bonds. Look at hemoglobin, very symmetric. Just a minute, you look at here. Suppose if I put 1, you also put 1 here. To this side, to this side. To this side, to this side. Okay? Next, here and here. Here and here. Here and here and here. So it is very, very symmetric and easy to remember. This side 1 and 1, 1 and 1, 1 and 1, 1 and 1, 1 and 1 and 1. So it is like if I am passing a plane, this half and this half are mirror images. Very easy to remember. And if I remove one such double bond, that becomes chlorine. Same way you have to draw, but remove one double bond. That becomes chlorine. Everything is same. So how many double bonds are in conjugation? Here 11. Here there are only 10. First two completed. Look at corrin. Any difference? Can you see direct linkage? This has been asked in many exams. There is a direct linkage between these two pyrrole rings. So you find only 3 methane bridges. Previously you found 4. 1, 2, 3, 4. For the porphine, for the chlorine also 4. But here only 3. 1, 2, 3. This is direct linkage. So this is a very important one to note. There is a direct linkage. Everything is a potential question. First you put 4 nitrogens, make them into 4 pyrrole rings. Pyrrole rings means immediately don't put double bonds. Just you put some alicyclic. Then you look at the double bonds. And in the first two cases there were 2 hydrogens attached to nitrogens. But in the next two cases only one hydrogen. 3 nitrogens don't have hydrogens. That is also important. Because if those 2 hydrogens if I remove, what will happen? If I remove those 2 hydrogens as H plus, then it will become 2 minus. Ligand will become 2 minus. So that is the reason magnesium enters into chlorine with magnesium 2 plus. Iron enters into hemoglobin with iron 2 plus. Because that is a dianion. 2 hydrogens are removed, dianion. Finished? Corphin is called as a puckered structure. Puckered means it will not be planar. First 2 will be planar. Next 2 not planar. Reason? Porphine will be planar. Chlorin, it is almost planar. Because porphine is aromatic. Aromatic has to be planar. But this one, which is having only 5 double bonds, this is non-planar. This is non-planar, or you call it as a puckered structure. Non-planar. Cyclohexane, is it planar? Cyclohexane. It is planar? No. Cyclohexane is in general not planar. It is puckered. And in this puckered form it will take up different conformations. It will take up chair conformation, boat conformation, half twist, twist boat conformation. They are important at symmetry level. In symmetry, generally they ask a question: what is the point group of boat shaped cyclohexane? Or what is the point group of chair form of cyclohexane? They have asked previously. You will wonder, B2H6 diborane, you are aware of this? B2H6, what is the point group of it? Not C2V. This was asked for 4 marks, part C. Just to know the point group, that's it. B2H6 point group, 4 marks. Hardly it will take 5 seconds. One time they have asked, what is the point group of allene? C2? D2D, this is right. Directly they have asked for 4 marks. You can go through all the previous CSR exam questions. Allene, D2D. So there are some molecules which you have to blindly remember. Allene, immediately D2D point group. Diborane, this belong to D2H. Ethylene, D2H. So these have been asked previously for 4 marks. So they are directly asked questions. If you are preparing symmetry, directly asked questions. Okay, note down the points for this. Just a few points. One example for each. Porphine. What is the difference between porphine and porphyrin? Because I mentioned it as porphyrin, but in brackets I have given it as porphine. Both are not same. This is actually porphine. In hemoglobin you don't find this. This is a base. This will have substitutions. 8 substitutions will be present. We call it as octa-alkyl substitutions. These are octa-alkyl substitutions, beta positions. With those substitutions, that will form hemoglobin, or that will form different hemes. Without substitutions, you call it as porphine. Remember, without substitutions it is porphine. With substitutions, that depends. It could be heme A, heme B, heme C, and so on. Substitutions will change. Okay? Write down. Porphine is found in hemoglobin. Porphine is found in hemoglobin, comma, myoglobin, comma, cytochromes. Hemoglobin, myoglobin, cytochromes, etc. etc. Next, since it is found in several proteins, comma, this macrocycle is called as workhorse macrocycle. Workhorse macrocycle. Workhorse macrocycle. What do you mean by workhorse? In industry, everyone should be like a workhorse. In pharma industries, workhorse means the one who works more, just like a horse. They have to work a lot. So in chromatography this question appears. Which is the workhorse chromatography? Workhorse chromatography. It means the chromatography that is used most commonly at industry level. Any answer? The chromatography which is most commonly used at industrial level. Thin layer chromatography, TLC. Once you put it on TLC, only then you will be going to the column chromatography or HPLC or GC, but every time you have to go for TLC. So this is called as workhorse chromatography. Likewise, whichever is found more, whichever is always functioning, whichever is always active, such things are called as workhorses. If you go to an industry, there is a project going on, 10 people are there, but out of those 10 people, all 10 people will not work efficiently. There will be only 1 or 2 who will be working more efficiently. They are called as workhorses. Okay, so in the macrocycles, which is found most commonly means porphine. That is why it is called as workhorse macrocycle. Okay, next point. Chlorin, it is chlorin, not chlorine. Don't put E. Chlorin is an element. Chlorin macrocycle is found in chlorin macrocycle is found in chlorophyll. In chlorophyll, where magnesium, remember magnesium not manganese, some students confuse between magnesium and manganese. In the options they generally give manganese also, and out of tension generally the answer will go to manganese. It is magnesium. Magnesium at the active site. Magnesium is found at the active site. Active site. See, these are the abbreviations, standard abbreviations. Even in the textbooks you will find hemoglobin abbreviated as HB, myoglobin abbreviated as MB, cytochromes abbreviated as CYT, chlorophyll abbreviated as CHL. Next, corrin ring can be found in coenzyme B12. Corrin ring can be found in coenzyme B12 and various forms of B12 and various forms of B12. Various forms of B12. Example, what is this? Not cyanocobalamin, that is why I asked. 70 to 80% of the times the students will pronounce this as cyanocobalamin. It is not cyanocobalamin, it is cyanocobalamin. Cyano you have to pronounce, then cobalamin, not cyanocobalamin. So don't break it here. Break it here. Cyanocobalamin or methylcobalamin or aquacobalamin, hydroxycobalamin. So whatever cobalt that is present here, different groups will be there. This group, if it is cyanide, then I call it as cyanocobalamin. Suppose in the place of cyanide, if there is OH, hydroxycobalamin. So even if it is hydroxycobalamin, what is the ring? Same corrin ring, it will not change. Ring will not change. Or if I put some other group, it is named by that, hydronium cobalamin or something else, aquacobalamin or hydroxycobalamin, cyanocobalamin, methylcobalamin. If I put methyl, you call it as methylcobalamin. This is very important. Any person who is deficient of vitamin B12, if you go to a doctor, what injections do they give generally? A cheaper one will generally contain cyanocobalamin, but costlier ones, it is like B12 only. If you go to a medical shop, you ask him for a B12 injection, one they will say about 30 rupees, one they will say about 100 rupees. Why one is more costlier? Because that will be methylcobalamin. This can readily give you B12. because once it goes, this gets metabolized. So this is a ready form of B12, therefore it will be more costlier, methylcobalamin. Okay, next one. Corphin ring is present in some nickel enzymes. Nickel cofactor, not enzyme, write it as found in some nickel cofactors. Nickel cofactors. So when I say cobalamin, which metal is present? This is second question. First question is cobalamin. Second question, which metal is present? Cobalt. Nickel cofactors, which metal is present? Nickel. Chlorophyll, which metal is present? Magnesium. Hemoglobin, myoglobin. Cytochromes, which metal is present? Iron. So two-way questions can be asked. These are hemes. I am just showing, you need not draw them. These are magnesium, chlorophylls. Some examples here I have given you of chlorophyll. And can you see? X is, if it is cyanide, cyanocobalamin. So the ring, there is a direct linkage, one linkage. Here you can see there is one methane bridge, one methane bridge, one methane bridge, but there is a direct linkage here. This is cobalamin. If X is cyanide, you call it as a cyanocobalamin. This one, chlorophyll. So chlorophyll, the ring that is present is chlorine, but you can see some substitutions. These are the substitutions. That will be in chlorophyll. But the porphyrin is a macrocycle. Base is porphine. The base is chlorine, but there are substitutions. That will become chlorophyll. These are some different heme proteins. This is, previously I told you, a cofactor. Where is nickel present? It is found in some nickel cofactors. Example, coenzyme F430. These 430, they are like absorptions. Now all these are not required for you. You are aware of all these things? Adenine, guanine, thymine and cytosine. Okay. So very quickly, this is not required theoretically. All the points are not required. But if I ask you a simple question, what are the basic principles of biological selection? It means, suppose, can you name a biggest scientist? One biggest scientist? Famous scientist? One very big scientist? Robert? Yeah, come up, no problem. Robert Hooke. So you are life science related? Okay, biology. Robert Hooke. The biggest scientist is nature. Nature is the biggest scientist. Yes or no? Because nature designs in such a way that no scientist can ever catch it. Till this point, every plant is splitting water molecule. So easily it is splitting water molecule and giving energy. So easily. But are we able to do it? Even after hundreds of years of research experience, no one is able to do it cheaply. They are doing it, but it is very, very costly. They are utilizing very high chemicals. Very costlier chemicals. But they are unable to make it cheaper. If it is cheaper, then I can go to sea, I can collect water, I can use it in my petrol bike. I don't call it as a petrol bike, water bike. Because if I can split water, whatever energy that comes, that energy can be utilized for running the vehicles. Plant is doing the same. In photosynthesis, when it splits water, what is happening? Energy comes out. That energy is stored. Okay? That energy is stored. So, nature is the biggest scientist. But whenever it is creating a species, maybe it want to create Shashikant or it want to create you, so on. Then what does it follow? Does it follow any principles or simply it will create you? It simply does not create. It will follow some guidelines. That guidelines, we have captured it. We have discovered the guidelines that the nature is following, and that we have given in the form of 4 rules or principles. What are these 4 rules or principles? Then the first one says rule of abundance. So in a very clear way I'll explain this, or in a very simple way I'll explain. I started an industry. Just imagine I started an industry. Okay, it is a pharma industry. So for my pharma industry, whichever are in abundant, I'll definitely take that. Whichever are in abundant. So organic, inorganic, physical, who are more abundant? Organic students are more abundant. So which one will I prefer? Organic, because say inorganic or physical students, hardly 50 or 100 students in the complete university. But organic students, I can get at least 4 to 5,000. So out of these 50 students, if I take all the 50, if one goes away, then how can I replace with an inorganic chemist? Difficult. I don't find inorganic chemist easily. I don't find physical chemist easily. But organic chemist, plenty of students. So my first rule is rule of abundance. Whichever is abundant, nature will first try to choose that. So when I come to elements, how many elements are there right now in the periodic table? 118. The last elements are anyway man-made. We call them as synthetic elements, transuranic elements. But the first elements are very common. The elements in the beginning are very, very common. So nature has chosen from those elements, from 1 to 30, most of the elements. After 30, very few. Why did not choose uranium in us? Why did not choose lead? Why bones are made up of calcium? Why iron is present? Why in the place of iron, why not ruthenium or osmium? Because they are group members, iron, ruthenium, osmium. Why iron should be present in hemoglobin or myoglobin? Why not ruthenium or osmium? Because they are rare. They are very heavier. They can't adjust easily. But iron can easily adjust. So compatibility, it checks everything. Nature checks everything, and it guidelines the preparation of a species. So the first guideline or the principle is rule of abundance. Whichever is in abundant, nature will choose it. Okay, here I've given you some shells, different shells. 90% of the shells will be to one side rotation, not to other side rotation. Did you notice that? Once they are rotating, they will be rotating to one side only. Suppose if I am starting from here, it will be rotating like this, from this view. It will not be like this. Nature maintains such a symmetry. You can see. So rule of abundance is the first principle. Okay, right. Now I got most of the organic chemist. Will I select all the 1000 organic chemists for my work? I will not. I will select those organic chemists from the abundance. I will select only those who are more efficient. Whoever can do the work efficiently, only them I will select. So the first rule is rule of abundance. Second rule is rule of efficiency. From those more abundant, I will choose only those which are more efficient. Okay, so that is called as rule of efficiency. Here we generally give an example. There are different electron carriers. Flavins are some electron carriers. Ferredoxins also are some electron carriers. So to carry electron, which is more efficient? Ferredoxins are more efficient. Therefore nature has mostly chosen ferredoxins, not flavins. Both are present in the nature, but why it has chosen mostly ferredoxins? Because they are abundant, they are more efficient. Why are they more efficient? Because metal will be present at its active site. In flavin, you don't find any metal at the active site. There is no metal at the active site. But in ferredoxins, there will be metal. We have to study ferredoxins separately. Flavodoxins and ferredoxins, flavin containing. So flavodoxin, here I've shown you flavodoxins. Flavodoxins are this. Flavin containing mononucleotides. Simply flavin. But if I go for ferredoxins, this is a structure of ferredoxin. Active site. But whenever we draw it differently. This is the what do you call protein synthesis. In that protein they will be giving all these things. So don't bother with these structures. We'll give you simple structures. This is ferredoxin, 2 irons, 2 sulfurs which are bridging, 4 cistenyl sulfurs. These are cistenyl sulfurs. Okay, so who is more efficient? Ferredoxins are more efficient. Flavins are abundant. Flavins are abundant. It means flavonoxins. They are abundant. Ferredoxins are also abundant. But who can do the work more efficiently? Ferredoxins. So it will employ ferredoxin. Flavonoxins to a very, very less extent. Okay. Third one is rule of basic fitness. What do you mean by it? Rule of basic fitness. You are abundant, you are efficient, but I want one particular thing. I want night shifts. But generally, do I employ ladies for night shifts? No, generally we don't. Or I have got R&D, I have got production. So where do I put ladies generally? In R&D, not in production. In production, I will be choosing gents because production will have more physical work. So though you are abundant, though you are efficient, but performing for one particular function, which one I have to choose? That is called as rule of basic fitness. There are some microorganisms. We should not call them as organisms because it is not organized. It is single cellular organism. We call it as single cell. These are called as diatoms. All these are diatoms, single celled ones. We use them in chromatography columns also for making the stationary phases. All these are diatoms, skeleton. They are like single celled. Usually these are eaten up by fishes and some other organisms. Generally they will be eaten up, but they have to survive. And for survival, they need a small protection. They make up a protection for themselves. They are single cellular, but still they protect themselves. How do they protect? They can prepare calcium carbonate, but calcium carbonate is opaque. It is like a solid, tough sheet. They they survive due to photosynthesis. But if they survive due to photosynthesis and if they prepare calcium carbonate, then they can't survive. They will die because light cannot pass through calcium carbonate. So calcium carbonate is abundant. Silica is also abundant. Silica, you are aware of silica, SiO2. Silica is also abundant. It will prepare a sheath with silica, not with calcium carbonate. Both are abundant, both are efficient. But for carrying photosynthesis, it requires silica. Because silica, at a very thin level, it is transparent. So sunlight can fall, photosynthesis can occur. So what is third rule? Rule of basic fitness. From the abundant, from the efficient, to carry out one particular function, whom should I choose? Nature will choose. That comes under rule of basic fitness. Examples I'll give you in the next class. And the last one is evolutionary improvement of efficiency. What do you mean by it? Efficiency and specificity also. I've given a simple example so that you will understand. How many of you are using this? How many of you are using this now at home? No one has it. How many of you have this? No. But this one? So there is an evolutionary improvement. You want to talk to your friend, you want to talk to your relative. Which one are you choosing? You want to improvise it every time. You are improvising. The same thing is also done by nature. At biological level, nature will also improvise every time. I'll give you a simple example of copper proteins. Generally, copper will have 4 coordination number. Copper will be surrounded by 4 amino acids at its active site. So for 4 coordination number, how many geometries are there? 2 geometries, square plane, tetrahedra. Moving from square plane into tetrahedra requires more energy transition. Because square plane is like this, tetrahedra is like this. So moving from tetrahedra into square plane requires great bit of energy. So every time copper 1 and copper 2, copper 1 prefers tetrahedra, copper 2 prefers square plane. So whenever it is undergoing a redox process, copper 1 into 2, copper 2 into 1, redox process. Copper 1 prefers tetrahedra, D10. Copper 2 prefers square plane. So it has to move from square plane into tetrahedra. This transition requires energy. So what nature intelligently does is, at any active site, you will not find these proteins in a perfect geometry. If it is tetrahedra, you don't find it in perfect tetrahedra. It will be distorted tetrahedra. If it is square plane, it is not in a perfect square plane. It is distorted square plane. So that from distorted tetrahedra, it can immediately go to square plane. If it is a perfect tetrahedra, from tetrahedra into square plane, it requires more energy. So it gets already distorted. From this distorted, it can easily go to square plane, or it can easily go to tetrahedra. That is the improvement that nature does. So that is what we call it as evolutionary improvement of efficiency, specificity. Okay, so these are the 4 principles that are followed by the nature. All these 4 principles and little points I'll give in the next class. Okay, and most importantly, in the next class we'll be taking up oxygen binding proteins, where most of the questions appear. If you take say 100%, nearly 70% of the questions you can see from oxygen binding proteins. So this we'll discuss in the next class. Okay.